Thank you so much. My name is Shaurya Agrawal, and today I'm going to present to you my project, a novel multi-stage cancer workflow using quantum and classical machine learning and microRNA signatures. So this project was mainly done at the Adlai Stevenson High School, along with some aid from the MIT Manol's Kels Lab. So first, let's talk a little bit about cancer and the limitations that we have with current cancer treatment. Cancer has caused over 10 million deaths in 2021 alone. It remains a leading cause of death, and any one drug class is ineffective on 75% of patients. So what this means is not drug or one or two drugs, but rather a class of drugs can be ineffective on 75% of different patients. And so this necessitates the need for tailored or precision medicine therapy. And currently what we see is that if you have cancer and you go to a doctor's office, they're going to do some simple tests. They might check your weight, they might check the origin of your tumor, do simple blood tests and simple biomarker tests. And oftentimes this leads to traditional therapy, which is one size fits all. And as you can sort of see here in this graphic, giving similar treatments to different cancer patients with different cancers might be effective for some patients, but it won't work for all of them. And so that means that we need to move towards precision medicine, where we look at the specific genetic and the genomic structure of the cancers and of what's going on inside the patient to give them better therapy that's tailored to their specific cancer and can lead to better treatment and better treatment across the board for various different cancer patients. And so even though current treatment methods are one size fits all, in the past decade, we have been seeing them move towards precision medicine. And essentially, there also remains an issue with the current precision medicine treatment methods that we have. And these precision medicine treatment methods use omics data. And that's sort of what's shown here on this image on the right. And on this image on the right, you can see how there's 5 different omics data types. And all of these omics data types represent a specific part of the cell of the tumor that represent a specific part of the genetic profile that the tumor carries. And this data is very hard to compute classically and very hard to simulate classically because this data is very high dimensional. So if you sort of think about it, sort of a rule of thumb is that each of these omics data types has at least tens of thousands of features associated with it, features meaning genes. And that's just because the human body has so many different genes. And all of these different genes combined together, integrated into one workflow, leads to this challenge here of data integration and feature selection. And having that many features and having that many genes in one data set is very hard to work with for classical computational systems. And additionally, this omics data is often very sparse and often very difficult to use and is also extracted from in vitro models. So what that means is that oftentimes these data types are extracted from lab cell samples and lab cell lines that are grown in the lab and don't really represent what's actually going on inside a human. And that's in vivo data. And so that can be very problematic and lead to low clinical applicability overall. And this is a crucial issue that needs to be solved. So then this leads me to the goals and needs of my project. The first is that we need a novel, non-invasive, and in vivo model of data for efficient and effective analysis. And the reason we need non-invasive is because, once again, omics data is extracted through biopsies. Usually the way we get omics data from humans or the way we grow lab cell samples is through extracting data from biopsies. And biopsies can be very painful, very invasive procedures. So again, in order for clinical applicability, we need non-invasive and in vivo models for efficient and effective analysis. The second thing we need is effective computational tools to deal with the high dimensional data that we see with omics data types. The third is generalizability to various cancers and drugs for higher applicability. And the fourth is an integrated end-to-end workflow for clinical applicability. And so this leads me to the proposed solution. The proposed solution is use of something known as microRNAs as our non-invasive novel potential biomarker extracted in vivo. So this will be the omics data type that we're planning to use throughout this entire project. The second would be an end-to-end workflow with 3 models to plan treatment for cancer. So that would be diagnosis, staging, and treatment. And the last would be a quantum machine learning model to effectively analyze the high dimensional and complex data of the treatment problem. So before we begin talking more about my project and essentially what we did, let's talk a little bit more about microRNAs. So what microRNAs essentially they're a regulatory mechanism for mRNAs. And maybe you've heard of mRNAs after COVID and the COVID vaccine. Essentially, mRNAs are a sort of vehicle. They convert genetic information from our DNA into actual proteins that have impacts throughout our body and that make us who we are. And microRNAs are a regulatory mechanism for those mRNAs after the transcription of mRNAs occurs. And essentially, this graphic on the left sort of shows here, you can see that this risk complex right here, the mRNA, the risk complex here represents microRNAs. And those microRNAs can either repress specific mRNA genes or it can completely cut out mRNAs. And so that means that microRNAs are a crucial part of this entire process of going from DNA to proteins, and the entire process that essentially leads to the ways our cells work. And so the reason we actually are using microRNAs for this project in general is because that microRNAs can be found as circulating biomarkers in the blood. So essentially what you might ask is that why are we using microRNAs when we can extract mRNAs, when we can extract DNA or actual protein information? The reason is that microRNAs can be found through non-invasive procedures, meaning microRNAs can be found as circulating biomarkers in the blood. And so now let's talk a little bit more about the machine learning aspect of this. So the machine learning model that we used in this project was a classical support vector machine. And essentially what SVMs do is they try to classify data by fitting a line or hyperplane through the data points. And you can sort of see in this graphic right here, in order to classify the green dots and the blue dots, the SVM essentially just fit a line through those data points to put the green dots on one side of it and the blue dots on the other side, essentially sort of classifying it. So like, for example, if it's given a new point that's on the left of the line, the model will assume that's a green dot, and on the right, it will assume it's a blue dot. And that's essentially what an SVM does for a simplified case. But that doesn't work all the time. And you can sort of see that here on this right image is that clearly the red and the green dots sort of form an oval shape. Like you need an oval shape to classify and sort of create that similar method of separating the data points. But an SVM can only fit a line. It can only fit a line or a hyperplane. And clearly you cannot fit a line or any hyperplane through this data to accurately classify it. You need a circular shape. And so here comes the kernel trick. And the kernel trick, essentially what it does is that it inputs this data into higher dimensions. It maps the input vectors into higher dimensions where it can accurately fit a hyperplane or a line through the data points. And that's sort of what's seen here in this parabola, in this three-dimensional graph, is that it took this data that can't be classified by a line and then mapped it into 3 dimensions where it can be through a hyperplane. And that's essentially what support vector machines do in the classical sense. So now we need to talk a little bit more about quantum support vector machines and what the theoretical quantum advantage can be. And quantum SVMs do everything very similar. They do a very similar process of the kernel trick of training the model and so forth. The only difference is that they use something known as a quantum feature map rather than a classical feature map. And the feature map in this case is just the algorithm that maps the data points from lower dimensions into higher dimensions where we can get that classifying advantage shown on the previous slide. And the reason a quantum feature map might have a theoretical advantage is because quantum feature maps work better with hard to simulate classically data. So like data that cannot be dealt with classically very well. And that's essentially what quantum feature maps can do. And the reason a quantum feature map can be effective is because it uses something known as the Hilbert space. So classical models can only transport input data vectors to so many dimensions. They can't go much higher because calculating the matrix for that becomes very difficult. And in this case, though, the quantum model uses the quantum Hilbert space. And the quantum Hilbert space can be classified and calculated much easier, and it uses infinite dimensions. So in this case, we get a better dimensionality we can get higher dimensions for these data vectors. Additionally, the quantum feature map can also provide an advantage in the sense that it uses something known as the ZZ encoding, which entangles the data in 2-qubit interactions. So the map can also use properties like entanglement and superpositions, as sort of seen with the header mark gates here, to extract the crucial interactions that go on between the different genes, between the different features in the data set. And that's something that's sort of needed with omics data and with mRNA data, because it's very high dimensional, it's very complex, and it's very highly correlated. So needing those higher dimensions, needing that interactions between the genes is crucial, and something the quantum feature map can provide an advantage for, and thus that sort of motivates the reason for using the quantum support vector machine. So the performance criteria essentially for this model for our workflow was, again, high accuracy, high generalizability, and high interpretability. So we just wanted those various different we wanted the model to be effective on those various different criteria and to be effective as proven against current literature. So here is our initial method and framework for our prototype. So after collecting the data and wrangling the data, after doing some initial pre-processing, we did some feature selection to choose only the most crucial genes, because as I sort of mentioned previously, these data sets can have thousands of genes, and that means thousands of dimensions, which is very hard to work with. So we just did some basic feature selection to reduce those data sets, or those to reduce those feature numbers into feature numbers that can be handled by our models. And so the first model, the diagnosis and the staging, the first two models use a classical support vector machine, because after some initial tests, it was sort of noticed that support vector classifiers, the classical ones, would do better than the quantum ones. And so that was sort of what was done for the diagnosis and staging. And then for the treatment, we actually use a quantum support vector classifier. So here you can sort of see the diagnosis model results. The diagnosis model had a 98% accuracy, and that accuracy was corroborated by all the other scores that we had. And these two graphs over here in the bottom sort of show the interpretability of the model. So the workflow not only has the three different models that we mentioned, but it also has something novel. It integrates those models. So the integration aspect comes from essentially that the diagnosis model, we extracted the most important genes, the genes that had the most impact on the model that led to the accuracy that the model had for the diagnosis and staging, and used those genes to form our data set for the treatment model. And that's why we need this interpretability over here. And so this left graph here sort of just shows how the genes were impacting the actual model. And as you can see, most of the genes actually had a negative impact on the model, and only a few of them had a positive impact. And even the ones with the most positive impact only had an impact of around 0.06. And that is much lower than our accuracy, right? Because our accuracy was 98%. It was nowhere close to being that low. And that sort of proves to us, along with this heat map, that the genes there weren't specific genes that had an impact on the model alone, but rather a combination of all the various genes. So we needed a lot of different genes to have this impact on the model that led to the higher accuracy. And so that's why we chose around 20 genes from this model to be inputted into the treatment model. Here we see the staging model results. So the staging model results were had very low accuracy, and so we did not really use this model for informing the treatment model. So that's why this model was really insignificant. And finally, we have the treatment prediction model. So the treatment prediction model had an accuracy of 76%, which was lower than our criteria of 80%, but it was still high enough, and it was corroborated by other scores. But a crucial result that we extracted from here was that the accuracy for a classical SVM was 73%. And after doing some statistical analysis, we found that the difference between the quantum SVM and the classical SVM was statistically significant, meaning the quantum SVM provided some sort of preliminary advantage over the classical SVM, the classical system that was seen. And you can also sort of see the interpretability of this model. So from the PCs, we extracted the most crucial genes, so the genes that had the most impact, the mRNAs that had the most impact. And we can clearly see that we have specific mRNAs that had the most impact and that led to the high clinical applicability for this model. So next, what we did was a redesign for the treatment model. So the treatment model had a 76% accuracy, which was lower than the criteria we wanted, which was 80% accuracy. And so we did a few redesigns for the treatment model. The first was an enhanced feature selection technique where we chose better feature selection techniques to better select the genes that we want that can have the most impact and not negative impact on the model. And we did an increase in PC and qubit dimensionality. So originally our model had only 2 PCs or 2 features. So we took all that information from the thousands of genes that we feature, or the hundreds of genes that we feature selected, and compressed it into just 2 features because each feature represents a qubit. And obviously we don't have quantum computers that can accurately or that can effectively use hundreds of different features and hundreds of different qubits. So that's why we compressed that into 2 qubits, but that led to a loss of information and the low accuracy that we saw previously. So we upgraded the model to 9 qubits and 9 dimensions. And lastly, the most important thing we did was quantum kernel alignment. So essentially, as I sort of previously mentioned, the only different part of a quantum model is the feature map. And the feature map here is oftentimes it can have a barren plateau. Like during the training procedure, there can be a barren plateau due to the dimensionality of the data, due to the complexity of the data. So what we did was we introduced a level of parameters. So just one layer of parameters before the data was actually inputted into the feature map for being transported into higher dimensions, into the Hilbert space. We trained parameters in order to fit the data better to the feature map so that it can be better transformed into the Hilbert space and into higher dimensions. And so in the redesign results, we see the treatment prediction model jumped up to 82%. And again, if you remember, our initial results for the classical SVM was 73%. So the treatment prediction model does seem to provide some sort of preliminary advantage in accuracy for this data type, or for these data types over the classical SVM. So after we do some interpretation, we can notice that mRNA signatures have high correlation and overlap for diagnosis and treatment overall, and mRNAs are an effective biomarker for diagnosis and treatment prediction. Additionally, what we noticed was that mRNA is ineffective for staging, and that's sort of already been proven in literature. And lastly, but most importantly, we noticed that quantum kernels can be more effective at hyperplane separation than classical kernels because of the fact if we use hard to simulate classically data. And in this case, we chose omics data. We use microRNA data, which can be very difficult to work with classically. And we do see some sort of preliminary advantage in terms of accuracy for this model. And lastly, we sort of see that the interpretation in gene ontology analysis, we see that the higher dimensional space for the quantum kernels was more effective, and we extracted crucial significant mRNAs that were found to regulate several different oncogene pathways and have impact. And so this sort of just proves that the quantum machine learning model also had higher interpretability, which is something that's not really been seen previously because these models can end up being end up to be black boxes. And so thank you for listening.
Okay, great. Thank you. We have already reached the time of your slot, but I think we can try to look at some of the questions because there were quite a number of questions here, down here. Maybe one of the questions was where did you have the data from? And,
sorry,
and what are the Yeah. So one of the questions was where did you have the data from, and what was the size of the data that you were analyzing?
Yeah. So I got the data from the TCGA data portal. It's a government available, it's openly available public data set where you can get a lot of different biological data and a lot of different clinical data as well for cancer patients across the board. So yeah, that's where I got my data from. And initially the model or the data set that I got, so the raw data set without any feature selection, without any pre-processing done, had around 1,100 features, so 1,100 different genes, different mRNAs that were included in the data set, but it only had around 700 samples. So it had more genes than samples than it had, and that can end up being harmful to the data set or to the model itself.
Yeah. Okay. Okay. Good. So let me just remind everyone that Shaurya has a channel in the Discord. So if you have any questions, then please direct him over there. Also, the links to all these things will also appear over there.
Definitely.
Right. I think I was going to have a question. So how do you think this problem is away from being used on an actual existing hardware?
Yeah, so I would say that to be actually used on an existing hardware, we need, I also noticed that there was often noise because of the increase in the qubits that I used, 9 qubits that I had. There was a lot of noise, and so a lot of shots were required and so forth. So I would say that we still need some time before we can get error correction and noise systems better in order to use these models, because even though we did see the high accuracy, there was a lot of time required. So that was the one drawback that we had, because classical SVMs can work very quickly. They're very fast models, but the quantum SVM was very slow because of the matrix that needed to be calculated, especially with such high dimensionality that it saw with the 9 qubits. So definitely there are some drawbacks that we see with the time and with the noise, but there was a benefit in the accuracy that we clearly saw that was statistically significant.
So are you saying that these results were actually computed on a quantum computer?
Yeah, so they were actually
okay.
Hardware, yes.